first thing we'll clarify two terms: autoreactive T cells or B cells and self-antigens. So any antigen that is coming from liver, from spleen, from lungs, from any body tissue of our own body will be called as self-antigen. And if this T cell is going to attack this liver, then it is going to be called as autoreactive, because it is reactive or dangerous against body own tissues. So just make it very clear that autoreactive cells are dangerous for our body, and self antigens are antigens from body own tissues, from liver, spleen, pancreas, kidney, lungs, skin. All these tissues are self antigens. Now, what is the main purpose of tolerance? What is the main purpose? So either they will delete them or make them non-functional. Okay, so they won't function. These are the two possibilities that are going to happen. This is the purpose of tolerance. So it will either delete the self-reactive T cells or make them non-functional. Now we'll straight go to first the T cell tolerance. So here I've just drawn the thymus structure. This is the cortex part of the thymus. This is the medulla of the thymus. Inside the thymus there are specialized cells. In the cortex, these cells are called as C is for cortical, T is for thymus, E is for epithelial cells, cortical thymic epithelial cells. So these two cells are cortical thymic epithelial cells. Below this white line is down here medulla, and these are MTEC, medullary thymic epithelial cells. Medullary thymic epithelial cells. And then these are the dendritic cells in the medulla. This is a blood vessel, and from this blood vessel the immature T cells will come here. So these are immature T cells that have come here. Now they are in the cortex. They are all without any CD4 or CD8. So even the TCR is under progress. So because these two receptors are not present on these T cells, that's why this phase is called as double negative. So they have no receptor on their surface, and they have entered from this bloodstream into the cortex of the thymus. So they are called as double negative. Now these cortical thymic epithelial cells, what they will do, they have got MHC on them, and with the help of MHC they will present the peptides. So in each direction this MHC peptide complex will be presented to these T cells. And what are the possibilities? There are three possibilities. Number one is that the T cell, it will bind very strongly. Number two, it will bind weakly. And number three is that it will not bind. Okay, so these T cells which are present here, these are going to come here, here, and here. And what they are going to do, either they will strongly bind, weakly bind, or they will not bind at all. The cells that will bind, strong or weak, they will be given signals to survive. So imagine this cell bind, this cell bind, and this cell bind. So they will be asked to live. They will continue further in the medulla, and this step is called as positive selection. And imagine these two, so these green ones, they bound, and this light yellow color, they did not bind, so they will die of neglect. Die of neglect means they are neglected, and because they are neglected, they are not given these survival signals. That's why they will die. Now, these T cells which have come here, they are all single positive. Either some of them will have CD8, some of them will have, so either they will have CD4 or CD8. Here CD4 and CD8. So now this is important. So let's say this CD4, this bound very strong. This will in the future become T regulatory cell. Okay, and these other cells will become normal CD4 cells or CD8 cells. So they started with double negative stage. There was no receptor on their surface. Then they came here and cortical thymic epithelial cells presented MHC along with the peptide. If they recognize MHC and the peptide weakly, then they will become CD4 or CD8 cells, one of them, and they will be given survival signals. This is called as positive selection. The ones which will not bind, they will die of neglect, and the ones which will bind very strongly, they will convert into T regulatory cells. So, 1 minute. T regulatory cells normally, what they have on their surface CD4 and CD25, correct? Now another T cell, simple CD4 cell, they also have CD4, and they when become activated, they also produce CD25. So what is the difference between this and this? What is the main difference? Most probably that will differentiate that this is this is T regulatory cell, and this is just simple CD4 cell. The presence of a gene called FOXP3. Okay, and this FOXP3, CD4, CD25, this together is called as T regulatory cells. Then T regulatory cells are of two types. One are natural Tregs or thymic T cell, thymic T regulatory cell. Another are induced I Tregs. Now these thymic or natural Tregs are these cells, and their selection started right in the cortex when they were positively selected by cortical thymic epithelial cells. So now we'll bring all these cells here. So here is CD4 cells. I'll just write 4 or 8 in the middle so it's not too much crowded. So 4 means CD4, 8 means CD8, again 4 means CD4, and this one I will write REG, regulatory. So they have now all come into the medulla. In the medulla, these medullary thymic epithelial cells, they have got a special gene in their inside which is called as AIRE gene, AIRE. And what is AIRE gene? AIRE gene is autoimmune regulator. Autoimmune regulator. And what it can do, it can create the expression of tissue restricted antigens. So for example, the antigen of liver, antigen of spleen, antigen of lungs, they all are tissue restricted antigens. And when these medullary cells, they express them here with the help of MHC, on their surface. This is called as ectopic expression. Ectopic now express. Let's say I will now write here L means liver, this means K means kidney. They will express now kidney antigen. Here S means spleen antigen. So this cell and this one is now expressing P, pancreas. They can express pancreas, spleen, kidney, lungs, any antigen on their surface, and that's why it is called as ectopic. Ectopic means it's not happening in the right place, somewhere else. The kidney is somewhere else, the lung is somewhere else, but its antigen is expressed by these cells. These cells express the antigen. Some of these CD4 and CD8 maybe will recognize, they are autoreactive. So let's say, I'll just mark them green again. Let's say this is autoreactive T cell for lungs. This is autoreactive T cell for kidney. This is autoreactive T cell for spleen. This is autoreactive T cell for pancreas. So when they bind to these MHC and spleen antigen, kidney antigen, lung antigen, pancreas antigen, they will be destroyed or deleted. So they will delete this cell, delete this cell, delete this cell, and this deletion is called as negative selection. Okay, and some of the cells which are okay, which don't recognize any of these, which just are normal, so they have the antigen receptor to recognize some other microorganisms, bacteria or viruses. These T cells will be allowed to go further. These T cells will be allowed to go as CD4, CD8 cells. Okay, so the first phase, where they just bind and recognize the antigen, and if they recognize strongly or weakly, these cells survive. This is called as positive selection. The cells that do not recognize anything, they die of neglect. Then those who have survived, they come to the medulla. Some of them are autoreactive against lungs, kidney, spleen, pancreas. They will be destroyed. That's why this is called as negative selection. And those who are okay, they have not recognized any self antigen, they will be allowed to go out of the thymus. And this phase takes about 4 to 5 days. The first phase take about 3 to 4 days. And so far in this negative selection and in die of neglect, 95% of the cells that enter, they are killed. Just 5% of the cells, they go out, they survive. Now another important thing to remember is that out of these cells, 1/3 will be autoreactive again. So this central T cell tolerance is not foolproof mechanism. There is still 1/3 of the T lymphocytes of this 5% which leave the thymus, which survive, they are still autoreactive. And to deal with these 1/3, there is further mechanism. And now I'll just briefly complete this Treg part also. So T regulatory cells which bound strongly, they strongly to the MHC and the peptide, they go further to this medulla. In the medulla, they also recognize the kidney antigen, for example, or the liver antigen. But they are the only cells that are not killed. They will continue as such their journey. Why? Because they will start expressing this FOXP3 and their role will change. They will no more be normal CD4, they will start to become T regulatory cell. So along this CD4, CD8, another cell comes out which is called as T regulatory. And because it is coming from thymus, that's why it is called as T regulatory or natural T regulatory.